Last February, the European Union awarded 2 gigantic grants to 2 big projects. They call them flagship science projects. Over the next 10 years, those 2 projects will receive each at least €500 million. One was about new materials, and the other one was about the brain, about neuroscience. It's called the Human Brain Project. It's hosted at EPFL in Lausanne, Switzerland, but it's a consortium of over 100 different institutions. And in very simplistic terms, what it wants to achieve over the years is to create a computer simulation of all the brain functions. Now, Sean Hill is the co-director of neuroinformatics for the Human Brain Project, and is also a leading expert in the simulation of large-scale simulations of brain circuitry. So it's a pleasure to have Sean Hill at the Brain Forum. Welcome.
All right. Well, it's a pleasure to be here. I wanted to start off this session talking about the Human Brain Project actually with a poem. And it's a poem about big data. And I bet you didn't know that such a poem existed. And it's a poem written by a woman in 1939. And she wrote, "Upon this gifted age, in its dark hour, rains from the sky a meteoric shower of facts. They lie unquestioned, uncombined. Wisdom enough to leach us of our ill is daily spun, but there exists no loom to weave it into fabric." I think that she really captured something that is very much the core, the essence of the big data challenge that we face today, and she captured this in 1939, so she really was ahead of her time. And in fact, in neuroscience, we're studying and we're gathering data from many different species, all in the aim of understanding the human brain. And of course, it's understanding the human brain that's very special to us. It's the human brain that underlies who we are. It's also at the core of many disorders and diseases that affect who we are. And the fact is that virtually none of these diseases or disorders do we truly understand. And none of the treatments that we have developed do we truly understand how they work. 70 to 90% of the drugs that have been developed do not work in 50% of the population. And the pharmaceutical industry, to a large extent, is actually decreasing or abandoning major new investments in the nervous system, in the developing central nervous system drugs. Now, one of the possible causes of that, although the regulatory issues are a big part of it, but of course the complexity, the cost is tremendous. So I think we need to think seriously about that, the impact of that, the approach to science, and large-scale brain initiatives to produce more data are being announced, it seems, every day. But the question is how to bring that data together and integrate it in order to advance our understanding of the brain. And that's really what the Human Brain Project is about. It's a 10-year initiative aimed at creating a global collaborative effort to understand the human brain and to enable advances in neuroscience, in medicine, and in computing. It consists of a consortium of 256 researchers from 135 research groups, 81 partner institutions in 22 countries, all around the world. And its objective is to really keep Europe competitive, to drive technological innovation while benefiting society. So the goals of the Human Brain Project are really to, first of all, lay the technical foundations for a new way of doing brain research using information and communication technology, to drive data integration and knowledge integration across multiple disciplines to better understand the brain, and to catalyze a community effort to really get everybody working together in order to develop new understandings of the brain, new treatments for brain disease, and new brain-like computing technologies. The brain is the ultimate supercomputer that we know today. At the core of the project is a scientific organization. I won't go through all the details of it, but the key point is there's a section here that's focused on producing data in mouse and in humans, but the core activities are really on building informatics-based platforms and then testing those platforms with specific applications. We know we're not going to map the entire brain in this project. Other groups are doing this, and we want to collaborate with them to be able to integrate the data that they're producing. But we do need some starting data. We need some key data to start fleshing out and building the scaffolding of these multi-level brain atlases that are necessary to start building the models of the brain. And the strategy to do that is really to start building what we call a data ladder to the human brain. For example, at the gene level, genetic level, at the transcriptome level, the proteome level, cell types, synapses, and identifying where is it that mouse and human are very similar, and where is it do they diverge. Because in the end, we're going to have to predict a lot about the human brain, and so we need to learn these principles and anchor it with a data ladder. So this is really a key point, and this is one that is often confused. The Human Brain Project is this process which is endlessly iterative, which means we're starting from data, we're starting from experimental data, from literature, from existing models, and using that to construct new models that we then run and evaluate on supercomputers, analyze the results, and use that to validate the models. And all through this cycle, this virtuous cycle, we're actually focused on continuing to integrate the data into this very detailed biophysical model that captures and is consistent with and has a place for all of the data that we've accumulated. So this is what we call a unifying brain model. And brain theory and mathematical theory, theoreticians are a very key part of this process. We don't believe for an instant that you're going to just take a whole bunch of brain measurements and throw them together in a bucket and suddenly understand how the brain works. These processes of data integration are guided across all levels by theory, by abstract models of the function of the brain, and this is always used to keep constraining and helping to guide the data integration process. So these platforms, there's 6 new platforms that we're promising to deliver, and we've got a tight time frame for delivering the first version of them within the first 30 months. And these are 1, neuroinformatics, brain simulation, medical informatics, high-performance computing, neuromorphic computing, and neurorobotics. And the intent is that these be open for the whole community to use. The first of these, the neuroinformatics platform, is actually designed to provide access and to federate all of the data from around the world, data repositories from around the world, make them accessible, and provide the tools to analyze that data, both structural and functional data, and to organize the data into multi-level brain atlases. And this means registering that data to standard spatial coordinates, using ontologies and semantic annotation to curate the data. Another key piece of this is to track the provenance and to build workflows. And this is really essential. If you're talking about a large-scale global collaborative project, you've got to keep track of where did all of that data come from? Who contributed it? Who contributed the analyses that were applied to it? Who contributed a model that was built from that? And these are some examples. What we talk about when we talk about atlases is really collections of data that are registered both spatially and semantically into brain spaces and brain vocabularies, brain ontologies. The brain simulation platform is really designed to provide an internet portal for neuroscientists to come in and say, I want to start building a model protein, a model cell, a model circuit, maybe a model brain eventually. Gives them the tools, it gives them the internet tools to start doing that, and then to run simulations, but instrumenting those simulations with virtual tools, virtual instruments like local field potential, or calcium imaging, or optical dye imaging, so that they can actually directly compare the results of these simulations to experiments that they can perform in the lab. And to link these simulations to a virtual body, so there's sensory input and motor output. And this is all in the aim of providing a platform for neuroscientists to really ask new questions using what we call in silico experiments. It also gives a tool to prioritize what is the most important new experiment to be performed. What's the most important key data that's missing and that needs to be measured in order to accelerate the progress in understanding the brain. So the challenge that we had was that there was 15 years of data that Henry Markram's lab had gathered, carefully using standardized protocols. So they had a tremendous variety of data. They had data characterizing the individual cell firing patterns, individual morphologies, synaptic patterns of communication, gene expression in individual cell types, the composition of cell types and densities, all for one single brain area. So this was all coming from postnatal day 14 rat, all in one area. So it was extremely well controlled, 15,000 experiments of data. And the question is, well, once you've done that work, once you've gone through the process to painstakingly gather all of that data, what do you do with it? And our challenge was to see, is it possible to take laboratory data and build a data-driven process that can automatically turn that into a model so that we can keep refining and integrating this data and know where to go next. And one of the challenges there was that there's a tremendous electrical diversity of cell types. Interneurons, for example, show many different firing types. And we don't know why. We don't know why there are so many different interneuron types, but we want to model these in order to be able to explore what they do. So we developed a technique that took those morphologies that were developed, took the gene expression information, took the model ion channels, and using an optimization process, a multi-objective optimization process, distributed those ion channel models across the morphology of the neuron until they recreate the firing patterns that have been measured in the experiments. And then we found that we could recreate basically all of the different firing types, and we could validate these models. They generalize to other types of stimuli than those that we built them on, including network-like input. So now we can build all the populations of the full diversity of neurons within the circuit, within this single cortical circuit. And to build the cortical circuit, then we use that diversity of morphologies, and we actually start placing those in 3 dimensions throughout the volume of the cortical circuit according to the composition that's been measured from the biology, and we start placing them. And there's, of course, we know a prerequisite for connecting neurons is that an axon come very close to another dendrite, otherwise they can't form a synapse. So the question is how do we wire this up? How do we get the positions of the synapses just right? How do we get them to match the biology? So we tried the process, we put them together, and the challenge that we faced was now we'd take a look at that circuit, we'd start flying into that circuit. And the point is that there are 2,970 possible synaptic pathways between the different cell types within this circuit alone. And only 22 of those had been robustly characterized. So the question is since those cost $1 million per pathway, and takes about a year per pathway to characterize, how are we ever going to be able to build this? So we needed to see, can we learn some principles to fill in that missing data? Can we predict how to assign these values? And can we predict the positions of synapses? So we had, from this 15 years of data, we had from painstaking pairs of reconstructed neurons that we knew were connected. We had distributions of the locations of these synapses. And we found that simply from the morphology alone, the shapes of the morphologies alone could predict most of the positions of the synapses. And it also pointed out that there are some exceptions. So there are some types of connections that we'll have to study further, and there are rules that we can layer in for those exceptions. But the vast majority, about 80 to 90% of these, were all predicted just by putting together those morphologies randomly within the space alone. This blew our minds. We didn't expect this at all. We thought we'd have to regrow the morphologies to get the right synaptic locations. But it means that the neurons are not growing to specifically seek one portion of another neuron. They're growing much more generally, and the synapses are formed at more stochastically selected locations. And we've learned that by layering in additional information, we can start to predict other features of the connectivity, including the numbers of synapses per connection. So for example, by layering in synaptic Bouton constraints, we can start seeing that we get the right numbers of synapses per connection, and layering in more rules, we're also able to start predicting for many of the pathways the probability of connection. Now we know on top of that, synaptic plasticity is acting, and it's shaping these individual synaptic collections. But when we put all of that together, we get network dynamics, these emergent phenomena that when we first put it together, usually when you build a network model, the first thing you get is you get epilepsy, you get seizures. But we didn't get that. By adhering to the data measured at minute levels, when we first put it all together and we activated it by simulating increase in extracellular potassium, which is an experiment that's often done in vitro experiments, we started to see these low-frequency oscillations. And in fact, that's the same type of phenomena that's seen in an in vitro preparation when you excite overall the slice using changes in potassium, for example. And we keep refining it every week, and we run thousands of validations across all different levels to keep improving this. And one of the things was just simply to make sure we have the synaptic minis, we've got all the right level of spontaneous activity within the slice, and then we could start to recreate experiments that apply more specific stimulations. And so one of those is actually some work done in Rafa Yuste's lab that stimulated thalamocortical efferents into the slice. And we simulated that with a density of synapses into layer 4. And what they saw is that when they stimulated those thalamocortical fibers at low frequencies, they got a little bit of a depolarization, a little bit of an activation of those neurons in layer 4, but it pretty much died out. And when they did it at a higher frequency, like 40 hertz, they actually saw this triggering of recurrent synaptic activity, and it spread throughout the slice. And here we are stimulating it at 20 hertz, and I don't know if the lights are sufficiently dim that you can see the details, but it basically depolarizes a little bit and then stops. And then, when we go to 40 hertz, and I hope you can all see this, you stimulate, and then it basically triggers this recurrent spread of activity, which actually follows the same paths of propagation that are seen in slice experiments. We keep seeing in which ways does it match the experimental data, in which ways does it not, and we use that to guide either getting new data or improving the model building process. So some of the things that are in process you heard about already, but a collaboration here with KAUST to model the coupling between the neurons and the glia and the vasculature, and this is in process. And then we're also working on scaling up this infrastructure to support modeling of the whole brain, so replicating that cortical microcircuit, and then we'll be working on all of the mesoscale and large-scale connectivity, fleshing out the rodent brain, and based on, of course, some key data in partnership with the Allen Institute and with other partners within the Human Brain Consortium. So the high-performance computing platform is really the platform dedicated to producing and ensuring that we have the compute power necessary to run these simulations. And a key part of that is that we need to have interactive visualization capabilities, because what we learned is that as these computers become larger and larger, and the direction that they're headed, they'll become so large that we can no longer write all the data from a simulation out to a hard drive. Just firing up those hard drives will cost so much and use up so much energy that it's no longer an option. And so what we're developing is we're developing this new type of application for supercomputers where you use them as an interactive tool to control the simulation and to visualize and analyze the results on the fly with the data still sitting in the supercomputer. And one of the challenges that we have, of course, is that we started off here with single neurons. This is the performance necessary to run simulations at these different scales of nervous system. And for a single neuron, it takes about a gigaflop, so 1 billion floating point operations. And then when we first started the project, we started with a single blue gene L, and we were able to simulate 10,000 model neurons. And then we've upgraded to blue gene P, and we've been doing about 100 times that. And then now we just recently took delivery on a new supercomputer that can get us to about the scale of a mouse brain Double that will be the scale of the rat brain. But we still, to get to the human brain, we need 1,000 times that of the rat brain. So that's an exascale supercomputer. And what's fortunate, and why this is an important moment in time, is that that's already on the roadmap to be delivered for about 2019. That's the year when the major supercomputer manufacturers are expecting to deliver exascale supercomputers. And we're in discussions with them already, making sure they understand our special applications. And they're very interested because this is different, it's something special, and it may be useful for more biological simulations. So they're making sure that their supercomputers can actually be used for these types of applications. Now, another key platform is medical informatics. And the goal here is really to federate clinical data from lots of different clinical databases, hospitals, and to make it possible to query that at an aggregate level. So not looking at individual records, but looking at an aggregate, as, say, for example, all of those patients that have been diagnosed with Alzheimer's. And then to look at their lab tests and to look at the details of their clinical data, analyze that as an aggregate, and see what are the subpopulations and how do we start diagnosing these diseases in a data-driven fashion and looking at the specific phenotypes of disease that emerge within that specific symptomology. So this is a new approach that is led by Richard Frakowiak, and we're working on deploying that first within Switzerland and then to grow from there. The neuromorphic platform is really about building hardware to accelerate and run these simulations very, very fast. And we're building off of 2 European projects. One is BrainScales, which builds analog VLSI, large-scale simulations on chips. And then Spinnaker, which is building off of the ARM processor, which all of you have in your cell phone, but using millions of them to simulate large-scale networks at real time. The neurorobotics platform is really about providing the simulated bodies, simulated sensory input, and motor output to couple with the simulations. And then some of the potential applications will be looking at the causal mechanisms underlying cognition, using this for developing new drugs for brain disorders, developing special controllers using brain-like principles. And we've got cross-cutting activities that are very important in ethics, maintaining a dialogue with society, with stakeholders, with philosophers, and with policymakers to make sure that there's a clear exploration of the ethical, social, and philosophical implications of the project. Educating, this is a key part, making sure that we're building and training the next generation of scientists to work on these types of problems, and to make sure that our results are widely disseminated with an open access policy on publications, data, software tools, and also the governance is a key part of this for ensuring collaboration across such a large-scale project. So I invite you to go to the website to look at the organizational chart. I'm afraid you won't be able to read it from where you're sitting, but there is 20% of the budget set aside for open calls. This project is designed and intended that we don't stay fixed with just the starting group of scientists from the beginning, but that it's open to participation all throughout the 10 years, and there's funding to support that. So thank you very much for your attention, and I'll be glad to answer questions later during the panel. Thank you.
Thank you.